Our work is to improve the delivery of photoimmuno conjugates to cancer cells through nanocarrier engineering. Before discussing photoimmuno conjugates, I'll first go over PDT. PDT stands for photodynamic therapy and requires two things. The first is a small molecule called a photosensitizer. Normally, this photosensitizer is non-toxic, but when exposed to light of the proper wavelength, it generates molecules known as reactive molecular species. These are toxic to cells and can kill them. The other aspect of photoimmunoconjugates is immunotherapy. All cells contain proteins called receptors on their surface. These receptors are critical for normal cell function and help the cell communicate with its surroundings by binding to molecules in its environment. In some types of cancer, however, the cells express certain receptors in numbers far greater than any healthy cell would. This provides an opportunity for targeted cancer therapy by designing molecules called antibodies that attach specifically to these cancer-associated receptors. Photoimmunotherapy combines these two distinct treatments. By chemically attaching photosensitizers onto a targeted antibody, we can deliver the photosensitizer only to cancer cells while leaving the healthy ones alone. We call this construct a photoimmunoconjugate, or PIC. When the cancer cell recognizes and takes in the PIC, the cell degrades the antibody, releasing the photosensitizer. When enough of this photosensitizer is exposed to light inside the cell, it becomes toxic and kills it. The biggest advantage of photoimmunotherapy, or PIT, is its specificity. By delivering the photosensitizer to only cancer cells, we can minimize damage to normal tissue when we do PDT. The main drawback, however, is that by delivering photosensitizer through an antibody, the amount of photosensitizer that the cancer cell receives is low, too low to effectively kill the entire tumor. So, there is a trade-off between the specificity and the amount of photosensitizer delivered, and hence the efficacy of the treatment when using this PIT approach. Our team's solution to this problem is to attach many PICs, usually around 40, onto a single central nanoparticle. Building on the hard work of others, we were able to successfully engineer the PIC and nanoparticle to allow for easy preparation and purification of this PIC nanoparticle construct. Using this approach, we hypothesized that we can increase the delivery of photosensitizer to cancer cells by increasing the amount that each receptor recognizes and brings into the cell. In order to make our PIC nanoparticle conjugate, we first mix each component in the proper proportions into a vial. The PIC and the nanoparticle are synthesized in such a way as to allow the formation of covalent bonds between the two, utilizing copper-free azide-alkyne click conjugation. This is allowed to react overnight. The following day, we purify the mixture using a technique called size exclusion chromatography. The size exclusion gel in this plastic column traps the smaller, unreacted molecules and allows the larger, conjugated PIC nanoparticles to pass through, which are collected as they exit the column. After purification, we characterize the purified product. UV-vis spectroscopy is used to measure the relative amounts of PIC and nanoparticle in our conjugate system. This is important for dosing calculations for treatment experiments. We also measure the size of the PIC nanoparticles using this instrument. It operates on the principle of dynamic light scattering, which is used to measure very small particles such as our PIC nanoparticle in solution. These two characterization techniques allow us to effectively characterize and determine the quality of our PIC nanoparticles. Thank you for watching, and I hope you enjoyed this video.